have a group of medical experts with important information regarding genetic testing.
Good evening, team. How are you all doing?
Fantastic. Excited to be reviewing information regarding genetic testing for cancer.
As am I.
Let's get started then.
As you all know, the chief from the Division of Medical Genetics has brought us together to discuss the drawbacks and benefits associated with genetic testing for cancer. Would anyone like to start by speaking on the current techniques for these genetic tests?
Sure. So genetic testing for cancer is relevant today because it helps determine if the cancer is due to an inherited gene mutation and whether or not someone is more susceptible to developing a second cancer. The process of genetic testing generally goes something like this. Hello, ma'am. Before we begin, I will need you to fill out a family history questionnaire.
A thorough questionnaire includes questions regarding the patient's personal and family histories of cancer. Some notable flags for predisposition to hereditary cancer include a family member with a known inherited mutation, cancer of the colon, uterus, or breast diagnosed under the age of 50, ovarian cancer diagnosed at any age, 2 or more close family members who have the same type of cancer, and the same type of cancer found in several generations of the family. Patients with a family history of cancer can seek genetic testing to determine if they have inherited dysfunctional cancerous genes. Each test is done in search for specific genes.
Now that that has been taken care of, I'll need to gather a body fluid sample. For this test, we need to take a blood sample.
Is it gonna hurt, Doc?
You won't feel a thing.
Youch!
Now, I'll send this sample to the lab and let them analyze it. We will call you in a couple of weeks to schedule an appointment so that we may discuss the results.
Okay, thank you so much.
The sample is then sent to a lab where the chromosomes and DNA can be analyzed. The results are reported to me or a genetic counselor. You will then be given the results during another counseling session. If the test result is negative, then it means that the gene mutation that was tested for is not present. However, it's also important to understand that the test cannot always guarantee that you're not at an increased risk due to any unknown genetic factors not tested for during this specific test. If the results are positive, then this shows that you have a mutated gene or genes. If the tests do show an increased risk, some ways to lower the risk may include taking medications or having preventative surgery, as well as making certain lifestyle changes.
That was a great overview of the testing process. Now, could you go more in depth as to how the tests are done within the lab?
In order to isolate the DNA from the extracted cells, the scientists generally mix the cells with some form of detergent in order to break down the outer lipid layer of the cell. Substances such as RNases and proteases are then added in order to dissolve the unnecessary proteins and RNA fragments. The sample is then treated with salt water and run through a centrifuge in order to separate the DNA from the leftover fragments and other materials. Then, in order to purify the DNA from the substance, many different techniques are used, such as treatments with ethanol, phenylchloroform, and or miniclone. After this is done, the DNA is treated with a DPA indicator to ensure the presence of DNA. Once the DNA is separated, the scientists are able to use computer software to analyze and compare the chromosomes to known genes in their databases to determine whether or not the specific gene/mutation is present. This patient had a family history of breast cancer and ovarian cancer, so I tested her for mutations in the BRCA1 genes. The BRCA1 gene is one of the most common genes tested for. This gene, when functioning properly, is a tumor suppressor, meaning it prevents uncontrolled cell growth by coding for products that either cause cell death or inhibit mitosis, therefore preventing cancer. When these genes become mutated, they stop working properly, and so they stop acting as tumor suppressors. This means cell proliferation can occur without being inhibited. It's like a car with no brakes. Mutations in these genes increase the risk for breast and ovarian cancer and are also associated with increased risks of additional types of cancer. BRCA1 mutations account for 20 to 25% of hereditary breast cancer and about 5 to 10% of all breast cancer. Other cancers that have also been linked to BRCA1 mutations include fallopian tube cancer and prostate cancer in men with significant BRCA1 and 2 mutations. Men and women with BRCA mutations are at a higher risk for developing pancreatic cancer as well. The BRCA1 gene codes for the BRCA1 protein. The protein is responsible for repairing certain types of DNA damage, and it prevents the cell from dividing until the damage is repaired. A person can have one bad copy of the BRCA1 gene from either parent. However, as long as there is one working gene from the other parent, BRCA1 will still regulate cells. However, there are hundreds of BRCA1 alleles, and some of them are better at repairing DNA than others. So even though a person may only have one dysfunctional BRCA1 allele, the other alleles do a better job of repairing damaged DNA. BRCA1 works by coding for the BRCA1 protein, which is activated by ATP. This protein will then phosphorylate p53, a protein product of another tumor suppressor gene. p53 will activate, bind to the DNA, and do one of two things. One thing p53 will do is transcribe the cellular machinery required to arrest the cell cycle so that damaged DNA can be repaired. If the DNA damage is extensive, then p53 will transcribe the cellular machinery necessary for the cell to undergo apoptosis. The problem is that when BRCA1 becomes dysfunctional, neither apoptosis or cell cycle arrest can occur. This means that DNA damage can accumulate and cause mutations in the cell, leading to cancerous growths made up of non-Vince cells. Yes, BRCA1 is an important gene when talking about breast cancer. These genes are tumor suppressors. However, cancer can also be caused by a dysfunction in proto-oncogenes.
Proto-oncogenes, like tumor suppressor genes, can cause cancer when they become dysfunctional. However, these are different from tumor suppressor genes because when these genes become dysfunctional, they will create an excess of mitotic products or prevent cell death. It's like a car with a foot constantly on the gas. RAS family genes are one type of proto-oncogene. 95% of pancreatic cancers and 45% of colorectal cancers are due to mutated RAS genes. This gene codes for the RAS protein, which plays a role in activating cell proliferation. A growth factor will land on a transmembrane receptor on the side of the extracellular matrix. The activation of this receptor will cause the phosphorylation of several other downstream molecules, which will phosphorylate RAS and activate it. From there, RAS can activate a protein, which causes a domino effect and phosphorylates downstream molecules that activate mitosis. It can also activate a different protein, which will phosphorylate different downstream molecules and ultimately inhibit apoptosis. When RAS genes become mutated, RAS will remain active without the signal from a growth factor landing on the receptor, meaning it's constantly activating mitosis while inhibiting apoptosis, causing an overproliferation of cells, aka cancer. Unlike tumor suppressor genes, which require 2 mutated copies of the gene for cancer to occur, proto-oncogenes only require 1 mutated copy of the gene. This means if one allele from one parent is mutated, but one allele is still in working order, cancer can still be induced. Any mutated tumor suppressor gene or proto-oncogene can be tested for with genetic testing.
Okay, so we've covered a basic review of what the types of cancerous genes we can test for. We should also talk about the issues that come along with the genetic testing for cancer. For instance, one of the main controversial points of view include a patient's right to not tell family members if they have cancer or a genetic predisposition for cancer. I've seen this arise with a few of my patients, and it's important that we give unbiased guidance when dealing with this sort of issue. Patients are sometimes hesitant on this matter because some test results may cause more anxiety for the person, especially if there's not much that can be done for the disease.
How will the results of this test affect my life?
If results are positive, you need to decide whether or not you want your siblings and/or children to know that they could be at risk for developing cancer. I'm not allowed to inform your family members of your results because I'm bound by HIPAA, the Health Insurance Portability and Accountability Act. It ensures privacy and confidentiality for all patients. You can decide if you want to inform them of your condition. If the test results are positive, you may want to take preventative measures, but it is possible for the test results to show a false positive. A false positive can cause unnecessary stress in your life if you are not actually predisposed. As with other personal issues, telling people is at your discretion. The Genetic Information Non-Discrimination Act, GINA, of 2008 was designed to protect individuals from discrimination in health insurance and employment. Genetic information in this context includes information about an individual's genetic tests and the genetic tests of an individual's family members, as well as information about family medical history. The law specifically forbids discrimination on the basis of genetic information when it comes to any aspect of employment, which includes hiring, firing, pay, job assignments, promotions, etc. Essentially, an employer may never use genetic information to make an employment decision because genetic information is not relevant to an individual's ability to work. Under GINA, it is illegal to harass a person because of his or her genetic information, Harassment can include making offensive or derogatory remarks about an applicant's employee information. The law does not prohibit simple teasing or offhand comments, but harassment is illegal when it is serious enough to create a hostile work environment. The law, however, does not cover life insurance, disability insurance, and long-term care insurance. There can be some controversy associated with this since some employees diagnosed with cancer are not covered with life insurance, and this is unfair to them. Unfortunately, mutation carriers may find themselves ineligible for life insurance and/or other long-term agreements. BRCA mutation carriers, for example, may be subject to rejection by families or communities and may be regarded as unsuitable partners. It is sad, but partners may not want to bear children with a spouse that will pass on the faulty gene. My ultimate role is to let you know that you have options other than what I would personally recommend.
When thinking about genetic testing for cancer, it is important to think about the benefits and drawbacks. One of the major benefits of genetic testing is having the chance to get a better understanding of one's risk for a certain disease. If a patient receives a negative result on a genetic test in families at risk for a specific cancer, then it may help relieve anxiety or uncertainty.
Likewise, a positive result can help make informed decisions about the future, such as taking preventative measures to help lower the risk. In addition, a positive result can help lead to an earlier detection of the disease, and that can lead to getting treatment earlier on. If a patient is already diagnosed with cancer, then genetic testing of the tumor can often help determine the prognosis and help determine which treatments will be most effective.
In the society that we live in today, money is a very important concept for most people. However, cancer does not select its victims based on the criteria of wealth and social status. Cancer affects all people, rich or poor. However, the poor folks often cannot afford to have genetic testing done and have little to no peace of mind as to what will happen in the future. This affordability is one problem that is sure to be magnified in the coming years as testing methods become more accurate and costs increase. Also falling under this issue would be the thought of gene patenting. Gene patenting normally allows the entity who discovers the gene to patent it, controlling the rights to study the gene, which typically lasts 20 years. Pertaining to gene patenting in the USA, it was perfectly legal and heavily practiced before June 13, 2013. As of that date, over 4,300 human genes have been patented, allowing those with the ownership of the patents to decide how the genes would be used both commercially and non-commercially. The U.S. Supreme Court later decided in the case of the Association for Molecular Pathology versus Myriad Genetics Incorporated that genes were not intellectual property and that they could not be patented because nothing new was being created. However, the case did allow the patenting of human genes that had been altered in a lab setting, since these alterations were not found in nature. Now, you may be asking, why is this a big deal? Up until 2013, different companies could possess the rights to different genes and therefore own the sole right to test for those specific genes. In this case, one company could not simply test for all of those possible cancer genes. This concept could lead to having multiple tests done to account for each known cancer gene, as well as an increase in price for each test, being that only one company has the legal right to test for the specified gene. This would ultimately lead to a person having to pay for multiple tests with the possibility of a price increase.
So what does the future of genetic testing look like, you ask? Well, without a doubt, genetic testing will play a larger role in cancer risk assessment, detection, and treatment in the future. Today, scientists estimate that the 3 billion or so letters of the human genome contain about 25,000 genes. The next step will be to identify these genes and learn what each one does. This genome map will allow researchers to identify cancer-related genes much more quickly. Currently, the Cancer Genome Atlas, TCGA, is in progress. Researchers are working to map the genes of cancer cells. Their main goal is to learn how those genes are changes to produce the cancer. The TCGA works by using an integrated network of hundreds of researchers across the United States and Canada to collect tissue samples from people with various types of cancers. This project will essentially allow us to understand the genetic basis of cancer and thus make genetic testing more useful in the future.